Hello, this is Jordan Lujan uh, from GCU from class Chemistry 530 Biochemistry, and this is going to be my presentation on glycolysis, glyconeogenesis, and aldolase. So here's a quick overview. We're going to look at the glycolysis pathway, the gluconeogenesis pathway, not as intense on that one. I'm going to look at aldolase in glycolysis and gluconeogenesis. Also going to look at the isoforms of aldolase and the aldolase B expression and regulation. So here is a very large uh, image of the pathway for glycolysis and the pathway for gluconeogenesis. And there's also a few facts here that are differences between the two. And I just want to look at these before I go into the more specifics of the two. Also, I have citations to images at the end of the presentation. So when we're looking at glycolysis and gluconeogenesis, you can think of them basically as opposites of each other. Glycolysis is the breakdown of glucose, and gluconeogenesis is the creation of glucose. And if you look, that's why this picture on the left has two parts. On the left side, you're going through glycolysis where it's breaking down, and gluconeogenesis is building up. So glycolysis is a catabolic reaction. I should say process, maybe not reaction, but process. And gluconeogenesis is a anabolic, more or less, type of process. Now, with these, most of the steps are actually very similar, and in addition to them being similar, a bunch of the reactions at different points are reversible. Um, However, there are a few different reactions that are specific to each of the two different processes. And so that the chart on the right, we can look at that and see, exactly what those differences are. So the top part of the chart says glycolysis is the set of reactions that convert glucose into two pyruvate molecules. So, and if you look at the bottom of the picture on the left, you'll see that there's pyruvate with 2 in parentheses afterwards, meaning that it makes 2 pyruvates. Also, with gluconeogenesis, you start with 2 pyruvates. And this process of glycolysis is important for uh, producing um, the pyruvate, which will then go into the Krebs cycle, and eventually you'll have the electron transport chain happen so you can generate ATP. It's the way we get energy in our bodies. So glycolysis is important. Gluconeogenesis is a little bit different, but that is something that will occur partially in the liver as well. So whereas glycolysis happens, everywhere, and it is responsible for producing the raw material for glucose, as it says in the chart. So I'm at the second points in the chart now. So with gluconeogenesis, it says the raw material is lactate, amino acids like alanine and glycerol. So that is something that's important as well for the gluconeogenesis. If you do have that lactate buildup, then it can eventually be converted back to pyruvate, which then can go through the process of generating glucose, which then, if possible, your cell could then break down the glucose and go through glycolysis, assuming that the conditions are right. So glycolysis, as we see, it says it occurs in the cytoplasm of all cells. And gluconeogenesis occurs in both the mitochondria and the cytoplasm. And if you noticed at the bottom of gluconeogenesis, when you have the two pyruvates, there's actually two reactions there with two different enzymes than what is used um, in glycolysis going to pyruvate. So in glycolysis, going from the phosphoenol pyruvate into pyruvate, you use pyruvate kinase. That gets rid of that phospho portion of the pyruvate. Well, if you look on the gluconeogenesis on the bottom left of the the bottom right of the left picture, then you will see that pyruvate will actually work with pyruvate carboxylase to create the oxaloacetate, which is used in the Krebs cycle. So that is why it says, for the gluconeogenesis, that's on the purple in the third point of the chart, it says occurs in both the mitochondria and cytoplasm, because that oxaloacetate is part of the Krebs cycle, or sometimes known as, the carboxylic acid cycle, things like that. Well, the oxaloacetate then has to still get to the phosphoenolpyruvate eventually, so you need the PEP carboxyl kinase to even push to that step. And then for a while you have some reversible reactions that use the same enzymes between glycolysis and gluconeogenesis. It's not until you get back to the top where glucose is where you have some different enzymes, and we'll look at that later on. So again, and I did mention this already, if we're looking at the fourth point in the chart now, the glycolysis is a catabolic process, gluconeogenesis is anabolic. Glycolysis is exergonic, gluconeogenesis androgonic. And the last point in the chart is that glycolysis occurs through 10 reactions. There's actually one step that is almost like a double reaction, if that makes any sense. I'll talk about it later. And gluconeogenesis says two essentially irreversible reactions in the glycolytic pathway are bypassed by 4 new reactions. And that is what I was mentioning, a second ago about the oxaloacetate and all that portion of it. But let's move on to looking at glycolysis. So at the top of this slide, I have what is essentially a long-form equation of glycolysis. In a introductory high school biology class, it might be explained that glucose C6H12O6 is broken to 2 pyruvic acids, or 2 pyruvate. Eventually it becomes pyruvic acid just because of the negative charge that's left on the end, so a hydrogen will come smack on the side. But yeah, this is the full equation that once you move out of a freshman biology class, you should know that there's NAD and NADH that is involved, ADP, ATP, and organic phosphate. All those molecules you would know that are involved. And you can see at each step where these are involved as well in this very nice picture. So step 1, going from glucose, a hexokinase will come in and make glucose 6-phosphate. So the phosphate's added on the 6th carbon of the glucose molecule. This is really important. This is not the most important step, I would say, but it is very important because this essentially traps your glucose in the cytoplasm. That phosphate does not allow the glucose to free roam out of the cell. So, and also from there, you can actually start this process. So once this process starts, you'll eventually, get your 2 net ATP produced by the end of glycolysis. Now here's the deal, that hexokinase in step 1, it actually does need an ATP molecule to function. You are sticking a phosphate onto glucose, so you do need that phosphate to come from somewhere. It's going to be ATP. So there is a input of ATP. Also, if you notice on step 3, there's an input of ATP. And this will also require a ATP to take a phosphate and stick it on the other side of what is now fructose 1,6-bisphosphate. And then from there, you will go into the aldolase reaction. and isomerase. Sorry. And from there, you can start moving on eventually to get to step 7, where at this point you're going to actually have two molecules. If you remember, the uh, glycolysis breaks apart glucose into two pyruvates, and before you get to pyruvate, it's broken into two molecules. So step 7, you have an ATP added on, or sorry, generated from an ADP coming and taking off a phosphate from one thing, and also you will have another phosphate taken off before you get to pyruvate. Well, that happens twice. It happens for each of your intermediary molecules. And so there's, in the red, you can see at the bottom it says steps 1 and 3 minus 2 ATP. That's the 2 you have to put in. Steps 7 and 10 you get plus 4, because once again it's the ATP generation twice. And so you have a net ATP of 2 generated. So this glycolysis pathway is very important for organisms that might have a lack of oxygen. So they cannot do aerobic respiration. With the lack of oxygen, they will produce, still be able to produce ATP, although at a very, very low level. However, because of the NADH, that can eventually be recycled and used. You can do the lactic acid process and all that stuff, recycle some materials, and eventually get to the point where if oxygen is then reintroduced at an adequate level, you can start doing aerobic cellular respiration at a greater rate. Now, if you notice at a lot of these steps, it has the delta G. So you can see some of these steps are going to be more irreversible and more spontaneous than others. So you can just see that in the picture. For example, that first step has a delta G of -4. So it's going to be one of those important steps. All right. And also the very last step has a delta G of -7.5. Now I do want to point out that those numbers can essentially be changed due to cellular conditions, but these are just some, I guess, accepted values. So here are the enzymes for each step in glycolysis. And I'm just going to briefly explain what the enzymes do. Most of them actually have very similar um, jobs, and you can just tell by the names. For example, number 1, hexokinase. Well, hexo meaning 6 because we have a 6-carbon sugar, and kinase. A kinase is a molecule that has to do with adding or removing a phosphate. Well, then 2, phosphoglucose isomerase. So you're just making an isomer, basically moving parts of the molecule around. Step 3 is the phosphofructokinase. Again, step 3, we are adding a phosphate group. Then 4 is the aldolase. This is the one that is a lot different. This is one of our most important ones because this actually breaks the molecule in half, basically. It breaks our fructose 1,6-bisphosphate in half into its 2 pieces. So then 5 is triose phosphate isomerase. So again, this is just making an isomer of one molecule into another. 6 is the glyceraldehyde 3-phosphate dehydrogenase. It is taking away a hydrogen, dehydrogenating. And this is the step that I was saying has almost like 2 steps to it, because the hydrogen is removed, and that allows for an oxygen to come and attack. Because if you notice, you'll see in the pictures, you can see on steps between 5 and 6, if you count the oxygens, you'll count slightly more oxygens. So I just think that's interesting. Or is it in this picture? I think this picture might actually leave it off, but there is a picture somewhere. You can find pictures of intermediary steps online. But let's move on to 7. Phosphoglycerate kinase. Again, that's a kinase, meaning it's going to move a phosphate group around. So we're going to get a phosphate group taken off in order to generate an ATP. Next, after that is step 8, phosphoglycerate mutase. A mutase is a form of an isomerase, basically. So I think about it, it's almost like the word mutation. It's, being changed. Even though it's not a mutation in the way of DNA, you would consider mutation. I'm just thinking kind of the colloquial term that you would see in, I don't know, a comic book movie or something where they talk about a mutation. You're different from someone else. Um, but I'm thinking of that more as a molecule. The molecule's being moved around. It's an isomerase, basically. Um, and then number 9, enolase. This enolase works pretty much as a dehydrogenase. Again, it's taking away a water group, and you can see that in the picture. And then 10, pyruvate kinase, our last one. That is, again, kinase. It has to do with a phosphate group. And we are finally left with pyruvate afterwards. So now the gluconeogenesis pathway. And we're going to start, this picture starts in the bottom right, where you can see the TCA cycle has the oxaloacetate there. Also, you have a pyruvate, and as I was saying previously, the lactate is could be a starting point to be converted to pyruvate, and then from there can go to the oxaloacetate. And so from here you can see our steps where we have the pyruvate carboxylase, so it's adding a carboxyl group to it. Now this is important because it actually requires biotin as a cofactor, and so you need that as well. The biotin is an important part. And a second reaction there, so this is only one reaction to get from phosphoenol pyruvate to pyruvate in glycolysis. And gluconeogenesis, you need 2 reactions to get from pyruvate to phosphoenol pyruvate. So the first reaction is the pyruvate carboxylase, and the second one is the PEP carboxyl kinase. So the PEP carboxyl kinase, this one actually requires a GTP instead of an ATP. Um, I think that's pretty interesting because almost all of our reactions for these two processes, this is the only reaction that does not have a ATP as far as a phosphate related thing goes. I just think that's really interesting. ATP or ADP, I should say. So the GTP to a GDP for the PEP carboxyl kinase into our phosphoenol pyruvate. So the other reactions here are mainly the same. They're just reverse direction reactions of what we had for the glycolysis until you get to the end of this process, which is going to be on the left side of the picture. And this is where you have your fructose 1,6-bisphosphate. So you have the fructose bisphosphatase that is going to put it to a fructose 6 P, and then another reaction to the glucose 6P. And so here is our last reaction that requires a new enzyme, which will be the glucose 6 phosphatase, which will remove a phosphate from glucose. And if you notice here, in a lot of these reactions, it's moving a inorganic phosphate, and it doesn't have it necessarily attaching to a ATP. So in this picture, so I think that's something interesting to think about. Also, if you notice halfway through this reaction, right in the middle, you have your NADH with your H plus, again being turned into NAD plus. So this is kind of what I meant about recycling if you need to go through this process after glycolysis. So let's look at what aldolase does. And this is from glycolysis. The aldolase is what is important for taking our fructose 1,6-bisphosphate and splitting it in half. Now, if you notice, it actually splits it into two different molecules. They are not the same. So you have your glyceraldehyde 3-phosphate, or the GAP, and you have your dihydroxyacetone phosphate, which is your DHAP. So these are essentially the same molecules in that they have same amount of carbons and things like that. However, in order for the reaction to continue on for one of those, you have to have your triose phosphate isomerase. So you need to change the isomer into something that can work with the reaction. So here we go. The Aldolase mechanism. In the top right, I just have a 3D molecule of Aldolase spinning around up there. It looks kind of cool. But so there's three main types of Aldolase we're looking at. Aldolase A, what these all really require is the lysine. The lysine residues are the very important parts, and I'll talk about that picture on the bottom in a second. But in Aldolase A, some of the important residues that are involved with it are tyrosine 363, lysine 146, and glutamate 187. So tyrosine 363 is a hydrogen acceptor, and lysine 146 binds covalently for stabilizing intermediates. Aldolase B, the important residue is lysine 229 and aspartate 33. And there is something called a shift-base formation that happens as an intermediary step, and that is the picture on the bottom I have shown. So we have our D-fructose, 1,6-bisphosphate, and our aldolase comes in, and you can see the shift-base intermediate that's pictured there, and eventually you get to your glyceraldehyde 3-phosphate, and the other one, the dihydroxyacetone phosphate, can be also the isomer of that can be made later on as well. So aldolase C, some of the important ones are glutamate 332 and lysine 71, and these form a salt bridge. So these are different. And from the papers I have listed there, even though they're not listed in the or as the sources on the slide, even though they're not, I don't have these facts listed. The aldolase, I think A and B are roughly 66 or 68% related. I think aldolase A and C are about 78% the same. So those are interesting. That's some interesting facts. Now here's a little bit of some of the difference as far as like the expression and things like that. So aldolase A is expressed in many tissues like muscle and erythrocytes, and the gene for this one is on chromosome 16. Aldolase B is expressed in the liver, gene on chromosome 9, and aldolase C is expressed in the brain and other nervous tissue, gene on chromosome 17. Now the picture I have in the top right there shows glycolysis. Aldolase A, B, and C are all involved in those. But also fructose metabolism, aldolase B can function in fructose metabolism. So that is very important, especially in the liver. Now here is some aldolase B gene regulation. There is a group of genes called HNFs, which are hepatocyte nuclear factors. So HNF1 is a strong transactivator, and HNF3 is a strong repressor. Now, what's funny about these two is they actually can bind at the same promoter region for the aldolase B gene, and HNF3 overlaps HNF1, so it can kind of keep it from, it can repress that from functioning as well. Now, HNF2 is a non-competitively inhibits DPB, which is on the next slide. I'll talk about what DPB is, but that's just an interesting thing as well. So DPP a D site of albumin promoter is what that means, and another gene, the CEBP. That's a hard one to say. Um, is a CCAAT enhancer binding protein. So the CCAAT is the cytosine, cytosine, adenosine, thymidine box motif. So in the protein that's a motif you would see that's just related to these type of genes. So the DBP is a strong transactivator. Um, the CEBP is not as strong in certain places, even though it can be expressed as well. The DBP is stronger. But these two function in highly specific locations in hepatic tissues. So specific hepatic cells are going to function better with some of these regulatory genes better than the other ones. So here are the picture sources, and here are the academic sources. Thank you for watching the video, and I hope you enjoyed.